into a vast look. Okay. And how do I make that go away?
It will go
I think
away.
it just goes away. All
Yes,
right.
and here you can.
And I can do.
They can
I don't know what that does. Okay,
it makes
cool.
them look at you when you're.
They don't have to look at me. I'm Mike Charleston. I've just gave a talk this morning, so I'm happy. I'm just chairing this third session. It'll run from 7 until about 8:30. We are missing one talk, and so after Constantine, his first talk, we'll jump to Chin Liu, and then we'll move on to Stephen Crotty, Sean Buckley, and then 2 papers to discuss, with brief introductions from Nick, whom we've already met, and Jonathan Mitchell, both of whom are here on campus, which is exciting. Kind of weird to be excited about being on campus, isn't it? You know? Anyway, so I'd like to introduce the first speaker. So Constantine is standing by, and I will add a note. I have read the abstract. I read all of the abstracts, and his abstract gave me the most work to do to get into the program because of all the bibliography. It didn't play nice. So totally irrelevant to his talk, which is going to be on something much more sophisticated, Bayesian phylodynamics, revealing the diversification process of Vanuatu's languages. So Constantine, could you start sharing?
Yeah.
He has. There it is.
Can you all see it?
Yes.
Okay. Then thank you for the introduction, and I'm sorry to make such an effort. And I realized that I was the only one who put references in the abstract. So sorry for that. Yeah, I'm a PhD student of Denise Kühnert at the Max Planck Institute for the Science of Human History in Jena in Germany. So good morning, Germany and Europe, and good evening, Australia. It's quite nice to give a talk at the end, at the other end of the world. So I will talk about Vanuatu and the languages of Vanuatu, and how we can use phylodynamics to get insights of the history of Vanuatu. So Vanuatu is also much closer to Australia than to Germany. It's a small island.
Oh, you want some baguette with peanut butter?
Say again?
I think everyone should check that their microphones are muted.
Okay. Yeah. Best is if you mute your microphones, then sounds the best. Okay. Vanuatu is a small island nation in the Pacific Ocean, northeast of Australia, which has few 100,000 inhabitants, and it features a huge linguistic diversity. So there were over 140 spoken languages on all the islands, and especially on one of its major islands, Malekula, there are 47 languages. So here you see a map of Malekula, which is like 40 times 30 kilometers, so really a comparable tiny island, and these are the boundaries of all the languages that are spoken there. So these are not only dialects, but these are really languages as different as English and French are. So the question is, how did it come to that? And the history of Vanuatu is pretty unknown to us because there is no written history there. We know that the first settlers arrived 3,000 years ago. We know that from archaeological records. And followed by that were many waves, several waves of immigration. So the first settlers came from the Austronesian expansion and spoke the Austronesian languages, which all the languages spoken today on Vanuatu belong to. And there were waves of immigration coming from the Papuan people, which brought their genetic content, but the people of Vanuatu kept their languages. So that was 3,000 years ago. What happened afterwards, we don't know really. We know there was a big volcanic eruption in 4052, which was massive. So it really had impact even in Europe, and definitely was devastating for the people of Vanuatu. And then we know that there were European colonists who arrived in the early 19th century and brought European missionaries, but they also brought the European diseases to the islands. And we see here a chart of the population size of Anatium, which is an island in South Vanuatu. And it is from 1860, it started with 3,600 people who lived there. And 1930 and 1940, it went down to 200. All European diseases, so influenza and measles, and a hurricane followed by sickness. So the 280 people we know that are living on Vanuatu today may have been 10 times higher. So the population may have been 10 times higher 200 years ago. So, but what happened in between? So 1850 is here and 3,000 years ago is here. We don't know really what happened in between. So we want to use phylogenetic methods on the languages to get an insight to that. The hypothesis is I want to test is where there are niche filling effects on Vanuatu. I mean by that is the islands were not inhabited before 3,000 years ago. So we could maybe see something like a lot of diversifications early in the time, or imagine something like a hurricane struck an island and killed all the people there. Then we again have an empty island which can filled, so the niche can be filled with new people and new languages can emerge. And this comes to the second point, what impact had catastrophes on the linguistic evolution? So we knew there were volcanic eruptions, hurricanes, and diseases. And can we see something there in the linguistic, on the languages that are spoken today there? So I used the Austronesian Basic Vocabulary Database, which is a database containing more than 1,500 languages spoken in the Pacific region. And all these languages come with a 210 item concept list. So I will explain what it is. It is, if you do phylogenetics with linguistic data, you usually have something called a Swadesh list. So that is a list with words that are most likely, or concepts that are most likely present in all the languages. So this is body parts, easy actions, and kinship terms, colors, low numbers, not something like tools or structures that emerge through cultural evolution. So for all these 210 concepts, which can be compared to like the core genome, if you think in genetics, a word is recorded that is spoken in this language. So we have the concept and then the word. And these words then are classified in so-called cognate classes. These are classes, equivalence classes of words that have the same meaning and share the same ancestry. So a simple example would be the English word hand and the German word Hand, or the Swedish word Hand. They all are considered as equivalent because they share the same meaning and they have the same ancestry, which is obvious in this term. But if you compare it to the Spanish word manu, which comes from the Latin word manus, it has a different ancestry that would then belong to a different cognate class. So all these words that are then classified in cognate classes are turned into a presence-absence matrix. So we have the word list for one language, and then you do a matrix, or which is a table for all the languages where all the rows are the languages and all the columns are the cognate classes. And we write a 0 if the language has not a cognate, and a 1 if the language has a cognate in this specific cognate class, and a question mark if we don't know. So this table here is then the alignment of our data. In Vanuatu, we have 326 documents collected in our Austronesian Basic Vocabulary Database. The problem is that I mentioned before that there are over 140 spoken languages, and we have much more data than languages. So there are many duplicates, which is at first a good thing because the more data, the merrier, but it has some drawbacks. So especially if you want to use phylogenetic methods. We are interested in the evolutionary, in the birth rates and the diversification rates, which are biased if we have too many data. So we have, especially on molecular, an oversampling. And the problem is it's not evenly distributed on all the islands. On some islands we have like 1 or 2 documents per language. On other islands we have like 10 to 12. The easy solution for that would be, okay, if we know what languages are spoken there, we can just choose 1 doculect per language. The problem is there's not something like an objective definition what a language is. But I need something to work with, so I came with a subjective definition, which is a bit arbitrary, but we will work with for it today. So I say 2 doculects belong to the same language if they have a shared cognate for at least 80% of their concepts. So if we have this list here for 2 languages, and we say this column matches for 80% of their cognates, then we say these belong to the same language. This is arbitrary, and the border is chosen so that the common understanding of what a language is fulfilled. So then I filter out all the languages or all the doculects which belong to the same language, so that only 1 doculect per language is in the data set. So if you think in trees, then you can imagine you have the full tree, which with all the data included, and then you filter out all the diversification events that are close to the present and have a filtered tree where you don't see diversification events close to the present. To get the tree from the data, the phylogenetic method is the same as for genetic data. We have our raw data and the cognate data and turn that into an alignment, which is the presence absence matrix I showed before. And then we need a substitution model that says what is the probability to go from a 0 to a 1 and from a 1 to a 0 over time, and a clock model and a tree model, which forms together the model. And we feed this into an MCMC search to get a posterior sample in a Bayesian way. So the posterior sample includes then posterior trees and the model parameters. To get an insight into the phylodynamics and how the history evolved, we especially look at the tree and the tree model. For that, I use the birth-death process. So a quick introduction. So we start, it's a process that generates a tree, and then we start at the root of the tree, and we have birth events, which are the green dots, where 1 lineage splits into 2 lineages, and extinction events, where a lineage goes extinct. And at present day, we make a sampling process where we sample a specific fraction of languages. So some languages are sampled and some languages are not sampled. We have several parameters that describe this model. We have the birth rate, which is the exponential rate that a birth event happens, and the death rate, which is the rate that a death event happens. And at present day, we have the sampling proportion. And we need additionally the age of the tree to compute the likelihood. We go 1 step further because we want to see the changes in these rates. So we use the multi-state birth-death model, where we allow that, where we define several states in the tree, and we allow for changes in these states. So we start with 1 state, which is defined by a tuple of parameters, lambda 1, mu 1, and at some point the tree might switch to another state, which has its own parameters. So this way we can detect areas in the tree which have a different dynamic. So here we have a simulated tree which has a lambda value of 1 in the red part and a lambda value of 10, which is much higher in another part. So we can clearly distinguish these two parts and see that in the model. So if we have some islands that have a completely different dynamics, we could see it in this model. So how does the tree of Vanuatu look like, of Vanuatu's languages? So we have a clear division in North and Central Vanuatu and South Vanuatu, which probably comes from the fact that there's a huge gap in between these islands. And we can see it in the tree. So we have two major clades, South Vanuatu, which has not that many languages, and North and Central Vanuatu. And even then, from all the islands, we mostly see that there's one clade per island. And the biggest one is Malekula, which is this island, which features also the highest linguistic diversity. So how do the rates look like in a multi-state birth-death model? We have now the net rate, so the birth rate minus the death rate. And red indicates a high net rate and blue indicates a low net rate. And we see that most over the tree, we have a fairly similar rate of 2.28 per 1,000 years, except of Malekula, which has a higher rate, and some areas of Malekula have even a really, really high rate. But I mentioned before that the problem is that we have oversampled Malekula, especially this area a lot. So this represents basically the amount of sampling effort we put in each island. So on Malekula, we have high sampling. On South Vanuatu, we have lower sampling. So I used the same approach to the filtered tree. So the filtered data set where I only have the 150 languages. And this is the result. So I basically see nothing here anymore. So all the rates are fairly constant throughout the whole tree. We maybe see slightly lower rates in South Vanuatu, but if you compare these values, they are close together. So we can say from that we probably have no islands that have a really different dynamics than others. However, there's one thing we need to know about extant time trees, because we have no ancient data in Vanuatu, and we need to be careful to infer the rates. So Stylianis Luca and Matthew Pennell published a paper this year demonstrating that estimating the rates in an extant time tree, so where all tips are from present day, is not that easy. The reason for that is that the birth rate and the death rate, they are highly correlated, and different scenarios, so different scenarios, how they can develop over time are possible. So the same tree can be explained with decreasing birth rate or an increasing birth rate. The rates, the absolute rates, can only be estimated at present day, where we have the sampling, if we know the sampling proportion. And one interesting thing is that the likelihood of the tree is not dependent on the topology of the tree. They show that, but only on the lineages through time, which is the amount of lineages that are present in the tree at each specific time. But they give a perspective to that. They introduce a reparameterization of these parameters. So let's go back to an extinct tree. So assume we have a tree with extinct tips, then we know from Stadler et al. That we can estimate these rates if we condition on one of those. So birth rate, death rate, and sampling proportion are correlated, but if we condition on one of those, so for example, sampling proportion, we can estimate the birth rate and the death rate. However, for extant trees, it's a bit more difficult. So Luca and Pennell introduced the concept of pooled diversification rates, which can derive from an LTT plot, inferred from an LTT plot. And if we condition on the sampling proportion and the death rate, then we can actually compute the birth rate from that. So I give a bit more detail, so that might be a bit technical. The pooled diversification rate is defined as the difference of the birth rate and the death rate, plus the relative slope of the birth rate. And they prove that the tree likelihood can be expressed solely in terms of the pull diversification rate and the product of rho lambda 0, which is the birth rate at time 0 at present day multiplied by the sampling proportion. And if we can express the tree likelihood only from the pull diversification rate, then we can estimate the pull diversification rate and this product by maximizing the tree likelihood. And the other fact is that we can compute lambda if we solve this equation for lambda. It's a differential equation, so we need the initial condition, and we can use the product lambda 0 times rho as initial condition. So if we know mu and rho, then we can compute lambda from the pull diversification rate. So how does it look like in Vanuatu? So we have the full tree of Vanuatu, so not the filter, but the full, and we calculate the lineages through time. And we see that it's quite an exponential curve, but it has, it's flatter close to the present. And we see that also in the log scale, that it flattens close to the present. From there, we estimate the pull diversification rate. So this is from the posterior tree sample, so that the thick line is the median and the blue area is the 95% confidence interval. And we estimate that not for the whole tree, because it gives quite quickly numerical errors if we have it on really small trees. So we start 1,500 years before present, estimating these pull diversification rates. And we see that it's fairly constant. So we see that the 95% error intervals, so that they get huge if we take really small trees, which is at this point really small. But they stay fairly constant, and we have a huge increase close to the present. And the estimated rho lambda 0 value is 0.35. So recap, this is the formula. What are the reasons why we see a high rate here? One could be that there's a really high birth rate, but the most likely thing is that we have a huge increase. So one thing to notice is that the pull diversification rate always looks backwards in time. So if we have high pull diversification rate, that means that the birth rate is probably falling forwards in time. And one other thing is that if this pull diversification rate is fairly constant, which we have here, then probably the rates are also constant. Because if the rates change, then this term would also be high and would not yield in a low constant pull diversification rate. So we have here the drop in birth rate probably, and here throughout the three constant rates. From there we can then compute the birth rates if we know the sampling proportion and the death rate. The problem is we don't know any of those. The sampling proportion, we can assume that it's most likely somewhere between 5% and 95%, but even more likely in 65%. We know that because we sampled all the area of Vanuatu, so we are quite confident that there are not much languages missing. But we don't know how many dialects are there or how many should there be in the sample. So we don't know at which scale of evolution we are looking. But a good estimate would be 65%. And we see that it doesn't matter much what we assume close to the present. It matters, of course, a lot. But the fact that it's fairly constant, the rates throughout the history here, is not dependent on the sampling proportion. It looks a bit different for the extinction rate. So here we have the birth rate, if you assume different extinction rates, and rho 65%. And we see that the higher the extinction rate is, the higher is the diversification rate. The extinction rate here is always assumed to be constant. And we see also here that the birth rate is just goes one step higher, the higher the extinction rate is, but it stays fairly constant over the time. The reason why we see this drop here is most likely because we still have diversified sampling going on. So we have a lot of samples on some of the islands, but on others we only have few samples which are purely distinct from each other. So it's not a random sampling, but a diversified sampling. And we can see from simulations that there also it happens that the birth rate then drops. So we cannot say much what's going on here because we have this diversified sampling going on, but we know quite confident that the rates are fairly constant before. So what can we learn from that? So were there niche filling effects on Vanuatu? Well, we cannot say what happened in the beginning because it's too far back and the confidence intervals are too huge there. But from 200 years to 1,500 years before now, we have constant rates. So we don't see anything like a boom event through a niche filling effect there. And what impact had catastrophes on the linguistic evolution? Well, we don't see any difference in the islands from the multi-state birth depth model. So all they are fairly constant or similar. So we have no visible impact on the evolution of basic vocabulary. So that's maybe one drawback, that we are only looking at basic vocabulary, which evolves very slowly and is not prone to borrowing. My interpretation for that is that the social dynamics of Vanuatu, so that's actually a quite interesting point. Why is that all also constant? And there are so much things going on in Vanuatu. And my interpretation is that the social dynamics of Vanuatu lead to a linguistic resilience. And that means that there was not something like a big emperor, which we had in Europe, which somehow then decided that all the people speak Latin. But there was a constant fluctuation which made a constant diversification of languages, which never got to a hold. And that's also a concept known from ecology. The recurring disturbances lead to stability. So because there were many things going on, like hurricanes, diseases, and volcanic eruptions, that all lead to actually stable evolution. So, but I'm more interested what you think, or I'm also interested what you think and what questions you have. I want to thank my group and the people from my institute, which also help a lot with collecting the data and preparing the analysis. And then I want to thank you for listening.
And then we go into this
And
one.
then we go into this
Camera
one.
control. Yay!
Right. Okay. Thank you very much. What a wonderful talk. Thank you, Constantine. I have a couple of questions queued because, you know, this is all very cool and invites questions, but I will step back. Does anybody else have any questions, and have they been in the chat?
We have a few compliments in the chat.
Compliments in the chat, well deserved.
I've got a question.
Yeah.
That was a really great talk. Do you try any like more standard coalescent approaches instead of birth-death models?
Yeah, thank you for the question. Coalescent processes always assume that we have a huge population and a small sample of the population. So that's one of the main assumptions of the coalescence process. And that is also the reason why we usually don't use coalescent processes in language evolution, because we don't have a huge amount of languages. We only have a few languages, and probably we have most of them in our sample.
Okay, thank you, Constantine. Any other questions
Yes,
in the chat? Yes,
Robin Ryder has raised a hand.
Robin.
Hi, yeah,
you-
thanks for lovely talk. Yeah, I was. It was very interesting. I was wondering about your rule for deciding which languages to prune out. So when you have languages which have at least 80% shared cognates, you keep only one. So first of all, how do you choose the only one that you keep? And also, you could imagine a situation where language A has 80% shared with B, who has 80% shared with C, but now A and C only share 64%. And so what would you do in that situation? And could you possibly try and get the cutoff automatically by using, I don't know, the kind of properties that you would expect from the pruning?
So the way I do it is that I have all the distances from all the languages to each other, and then I take the lowest distance that I see and throw out the language that has the fewer data. So that might be a quite pragmatic approach. And then I raise the threshold and go to the next pair of languages that has the lowest distance and throw out the one with the fewer data. And I go on until I'm at the 80% or 20% then from their distance.
Great. Thank you very much. Thank you, Robin, for your question. There are a couple more questions in the chat, and I have my two at least to ask as well, but I suggest we hold those off until the discussion towards the end, and then move straight on to our next talk. So before we do that, let's thank Constantine once again for a wonderful first presentation of this session. Really loud on the microphone. Next is Chin Liu, who's a PhD student here at the School of Natural Sciences at UTAS, and is zooming in as we speak. Chin, are you ready to go? Great.
Can you hear me?
Yes.
Can you hear me?
Yes, we can.
Yes.
Just trying to find my spotlight. Hello, everyone. My name is Ching. I'm a PhD student in the University of Tasmania. Today, I'm going to talk about model selection in phylogenetics. My topic is AIC an appropriate metric for comparing partition models and mixture models in phylogenetics? So AIC is an archaic information criteria. It is an asymptotically unbiased estimator of the relative expected Kullback-Leibler distance. AIC is expressed as -2 times the maximum likelihood of a model, given the data and takes away the number of the free parameters. The lower the AIC, the better the model. Because of its simplicity, AIC has become a very popular tool in phylogenetics. Some recent papers have questioned the effectiveness of AIC in phylogenetics, but there has not been any investigation of mixture or partition models. So we are interested in whether it is appropriate to use AIC to compare these models. So partition and mixture models apply multiple models to a multiple sequence in two different ways. To apply a partition model, the data is divided into some partitions, and then a model is fitted into each partition. A mixture model fits more than one model to each site in the data. Partition and mixture model can be used to accommodate heterogeneity across sites in the patterns of gene sequence evolution. In last year's Philomania meeting, if you remember, Stephen Crotty presented a talk. He investigated the performance of the mixture and partition models. He found out that AIC seems to always choose partition models over mixture models. Even the trees inferred by the mixture models were correct and partition models got a tree wrong. This is one of the motivation for our work in the investigation of AIC in partition and mixture models. So when the sample size is large, AIC is an unbiased estimator of the relative expected KL distance. It will be worthwhile to compare AIC to the EKL. Maybe one of the reasons of partition models always having lower AIC values than the mixture model is the bias of AIC in estimating EKL. Maybe AIC is more bias distant in EKL in partition models than in mixture models, and the bias of AIC can result in selecting the wrong model. So we are also interested to see whether the model chosen by AIC recovering the true tree topologies. Did the model having the lowest AIC values always get a tree right? If it is not the case, maybe we shouldn't just rely on AIC in model selection. So to answer this question, we set up some simulation. The first step is to create some heterotachously involved data. That is, data that sites may differ in either their rates of evolution or in the pattern of substitutional rate changes. Then we can fit some partition and mixture models into the data and evaluate the performance of these models. So the data set was generated under a 2-block partition model. Each partition includes a GTR model and a tree. These trees have the same topology but different branch length. Under each model, we generated 1,000 sites in CGen. Then we created some concatenated data set called 0%, 5%, 10%, and all the way up to 50%. So for 0% data set, we just simply join these green and pink sites together. For 5% data set, we randomly selected 5% of green sites and 5% of pink sites and swap them around. So each partition contains 5% of incorrectly partitioned sites. For the rest of the data set, we did the same thing as in the 5% data set. So we repeated this process 100 times. This gave us a total number of 1,100 data sets. So for the 0% data set, the true model is a 2-block partition model because it has no incorrectly partitioned sites. If we fit a 2-block partition model and a 2-class mixture models into this data, not surprised, partition models are going to do better than mixture models. But what about if we fit partition models and mixture model into this data set with increasing incorrect partition sites? And we evaluated each data set in IQ tree and recorded the Robinson fold distance to the two trees, AIC and EKL. To calculate the EKL, we need to use all side patterns as all possible outcomes. Due to the time limits here, I won't explain it in detail. If you're interested, I can show you the formula after the talk. And we conducted a few simulations, but I will only talk about the result from one simulation today. These are the two trees that were used to generate the data set in the simulation. They have the same topology, but different branch length. The reason to choose this tree is that in reality, we may find some data with some sites, not only having different rates of evolution in alignment, but also having different substitution rates across all lineage. So we wanted to set our trees with same topology, but different branch length. These trees have short internal branches, and they're kind of opposite to each other. For example, tip 1 has a longer branch in tree 1, but a shorter branch in tree 2. So for this simulation, we fitted 4 different partition mixture models. This includes 2 different partition models and 2 different ghost mixture models. Ghost is short for general heterogeneous evolution on a single topology. This model is implemented in IQ tree. The ghost model we use here is a 2-class model, mixture model, and both classes have the same tree topology, but different branch length. These are the total number of free parameters in the model. So this is the RF distance result. The right figure shows the proportion of the times a model recovers the correct tree topologies, RF equals 0. You can see that the green mixture models seem to have the highest proportion of the times in recovering the true topology than the other models in the simulation. It is quite interesting here for 0% data set. The true model is the blue partition model. So we would expect the blue partition model to perform the best in the 0% data set, but both the blue partition model and the green mixture model has the same proportion of the times in recovering the true topology, 70%. As the incorrectly partitioned sites increase from 5% to 50%, this green partition model performs better than blue and red partition models. And even the orange mixture model seems to perform quite well compared to both blue and red partition models. The left figure shows the mean RF distance for 4 different models. The green mixture model have the lowest mean RF distance. That means the topology of the trees inferred by the green mixture models are more accurate than the ones inferred by the other models. Again, for the 0% data set, the blue partition model is a true model, but the topology of the tree inferred by these green mixture models are more accurate than the ones inferred by the true model. This is the EKL results. The left figure shows the mean AIC value and the EKL values. The lines are mean AIC, dots are EKL. It seems like the mean AIC value do not agree with the EKL values. Based on the EKL values, the red partition model and the green mixture model are doing better. But if you look more closely, overall, the green mixture model is doing the best as the proportion of incorrect sites increases. But based on the mean AIC value, the red partition model seems to perform the best. For the 0% data set, the true model is the blue partition model, but EKL didn't choose the blue partition model for this data set. And in fact, the blue partition model performed worse among all the other models. The right figure shows the bias of AIC in estimating EKL. In this simulation, in general, AIC seems to underestimate the EKL. And it seems like the amount of underestimation is larger in the model with more parameters, the blue and the orange, than in the model with less parameter, the red and the green. So before I move on, I want to talk about what the best models are according to AIC. AIC as a value by itself is meaningless. We need to compare the AIC of a model with other models. We define delta AIC as the difference in AIC values between the model and the model with lowest AIC. And based on the delta AIC cutoff values, we decide which models are the best. For example, here are 4 models, ABCD, given a data set. These are their AIC values. And model A has the lowest AIC values. Then the delta AIC value for each model are here. If we choose a delta AIC cutoff value as 0, that is only the model with the lowest AIC values are the best model. Then the best model here is A. If we choose 2, the best models are A, B. So notice here, if we choose the maximum delta AIC values as the cutoff value, which is 10 here, then all of the 4 models are the best model. There's a lack of clarity in the literature in phylogenetics regarding what exactly defines the best model. According to AIC, the widely adopted rule of thumb for delta AIC cutoff value is 2. This figure shows the proportion of the times a model was chosen by AIC for 0%, 30%, and 50% data set. The x-axis represents the delta AIC cutoff values. As you can see, no matter how many incorrectly partition sites are in the data, AIC always chose the red partition models. Very close to 100% even when the delta AIC cutoff is 0. That is, nearly all red partition models have the lowest AIC values. It is interesting here for 0% data set because the true model is the blue partition models, but AIC actually prefer the red partition model more than the blue partition model until we reach a delta AIC cutoff values around 20. Another interesting here is that in general, AIC prefer overcomplicated model, but in this simulation, the red partition model has the lowest, has the smallest free parameter among those 4 models, 26. So AIC chose the simplest model in this simulation. And remember that based on the EKL values, overall, the green mixture model performed the best. And based on the RF distance, it is also the green mixture model performing the best. So the model chosen by AIC are not the same as the one chosen by EKL and RF distance. Now we want to know whether the model chosen by AIC recovered the true topology. This figure shows the proportion of the times model chosen by AIC recovering the true topology for different data sets, 0%, 30%, and 50%. First, we notice in this figure, all the lines seem to eventually approach different constant value as the delta AIC cutoff value increase. It is because as the delta AIC cutoff value increase to a certain value, all models are considered to be good model by AIC. So the proportion of the times all model chosen by AIC recovering the true topology is equal to the proportion of the times a model recover the true topology out of all the model, which is a result of the RF distance here. So for the 0% data set, the proportion of the times a model recovering the true topology is 70% for the green and blue, 0.69 and 0.67 for the red and orange mixture model. So these are the value of here for the 0% data set. And the same thing happened to 30% and 50% data set. So these regions indicate the chance of a model getting the topology right if I just pick a model randomly out of all the models. Let's call these values expected values. The red partition model is always a straight line here because as we noticed before, nearly all partition models had the smallest AIC values. And by looking at the expected value of the red models, not all red partition models got the true topology right. So the model chosen by AIC not always recover the true topology. Let's have a look at the models, blue, orange, and green. Before they approach to the expected value, we notice that this line segment, some of them are actually below the expected values. That means the chance of a model chosen by AIC getting the true topology right is lower than if I just pick a model randomly. So if our goal is to pick a model which recover the true topology, AIC fails in this selection criteria. Let's look at the result from the EKL again. EKL prefer the green mixture models, and the RF results show that green mixture models have the highest chance of getting the topology right. So EKL chose a model that have the highest chance to recover the true topology. Perhaps we should just use EKL or use EKL as another metric in model selection in phylogenetic if possible. So is AIC an appropriate metric for comparing partition model and emission models? Our simulation study suggests that the answer is no. There are two reasons for it. First, when the model parameters are near the boundary, AIC is a biased estimator of EKL even when the sample size is large. And the model chosen by AIC did not agree with the model chosen by EKL. The second reason is the chance of a model chosen by AIC recovering the correct topology is not always higher than the chance of a model chosen randomly recovering the correct topology. Thank you.
Thank you very much, Chin. Can you hear me?
Yes.
Yes, right. Are there any time for, yes, a quick question from Jonathan.
Firstly, that was a really good talk. I'm wondering if you have looked at the BIC.
No, I haven't. We just focus on AIC.
Yeah, so the reason I say that is because, so with the AIC, it's not actually a consistent in the sense that you won't necessarily recover the generating model.
Okay.
So, whereas the BIC is. So that's not like, that doesn't mean you shouldn't use the AIC, but if you want to recover the generating model, then it might be interesting at the BIC as well.
Yeah, I noticed, are you going to have a talk about BIC later on?
Yeah.
Yeah, yeah, we haven't looked at BIC, maybe it's worth to have a look maybe in the near future.
Yep.
Thanks for the suggestion.
Cool. Thank you very much for the question too. Let's thank Chin once again for a great talk. Right, so I saw Stephen Crotty on a screen. There we go, thank you very much. And I think you have a helper with you this evening, Stephen. Stephen is going to...
It remains to be seen if she's a helper.
He's going to talk to us about heterotacky mapping. We've heard a little bit about heterotacky, is that heterotackous? Yes, models, and now we're going to have a little bit more. So again, this is a 15-minute talk with time for a question or two at the end. Thank you.
Okay, so good afternoon, good evening, good morning, everybody, wherever you are. Let me just see if I can figure out how to make my slides go full screen. So is that, everyone can see my slides here?
Yep.
Yes.
Okay, excellent. So firstly, I think I need to immediately thank Jin for the last talk because it fills in a few gaps that I left in the kind of the introductory part of my talk. So I did actually change the title that I put in the abstract from heterotacky mapping to phylogenomic signal mapping, which, yeah, I may or may not explain why as we go through. But Chin explained a bit about the difference between sort of petition models and mixture models, which I'm just going to very briefly sort of tell you the way I think about it. If we have a set of sequence data here, and let's pretend that it's two genes, a petition model, traditionally we would say, okay, well, there's two signals here we're trying to recover, two sets of parameters we're going to recover, and one set is going to be with gene 1 and one set is going to be with gene 2, okay? A mixture model, we can also say, well, there's two sets of parameters and two signals we're trying to recover, but we're not going to assign one sort of class, as I've called them here, to one gene and one class to the other gene. We're going to say, well, there's two signals, but they sort of apply in some unknown weight across the whole data set. So the ghost model, as Chin explained before, basically is implemented in IQ-tree, and it allows allows you to infer a number of different classes, so 2, 3, 4, however many you think you need. So each class has, you know, a different set of branch lengths. And And so that's all I'll say for that. I think it'll hopefully become clear. What I wanted to explore is heterotacky, and effectively that's kind of this sort of thing when I guess evolutionary pressures or forces change at different times throughout history or different species along the tree, you end up with this weird mixture that we're going to try and recover. And I wanted to do some simulations where we had some idea of the strength of the heterotacky, which I haven't actually done before. So I did something which is really simple and most certainly not biologically plausible, but it's nice to be able to measure it. So what we've got here is just a random tree with 14 taxa, and we have two distinct branch lengths. The long branches you can see are of length 0.75, and the short branches you can see are of length 0.05. So all the branch lengths are either 0.05 or 0.75. All right. And I'm going to say that, okay, this is a tree for class 1 if I'm going to have, two different signals. Now, the amount of heterotacky that we're going to have, if we have no heterotacky, then the tree for class 2 is essentially the exact same tree. Okay. And I've coined this term heterotacky factor, which is going to relate to kind of the strength of the heterotacky going from 0 to 1. So if there's no heterotacky, well, the tree is exactly the same. If we want to introduce some heterotacky, the heterotacky factor is essentially the number of branch lengths, or the proportion of branch lengths in the tree that flip. So what I've done with, and this is actually, it was my OCD that led me to choose 14 taxa, because that gives you 25 branch lengths, which is a nice round number for giving you nice even proportions. So a heterotacky factor of 0.04 means that there is just one branch out of the 25 that has changed. And you can see it's this one here. So in the first tree, this was a short branch. In the second tree, it's a long branch. Now, that one branch that changes is selected randomly. So it could have just as easily been a long branch selected here that became a short branch here. All right. So that's basically a little bit of heterotacky. The trees are basically the same except for one branch. What have we got next? Okay, so if we have this tree, and what have we got? 0.4. So this is 40% of the branch lengths have flipped. So 10 of the 25 branch lengths have changed. If they were small here, they are big here. And if they were big here, they are small here. And the 10 out of 25 are selected completely at random. All right, so you can see now that these trees start to look quite different. And if we take it all the way to the extreme with a heterotacky factor of 1, that means all of the branches are different. The short branches are long and the long branches are short. And you can see they look quite different. So now this is where I'm going to start sort of waving my hands and hoping you can kind of follow and work out what I'm talking about here. And it would have been nice to this kind of follows on from my talk from last year. So those of you that were there and have a good memory might pick up something here. But what I'm actually looking at here is, let's say we've taken one of these simulated data sets, and this is one where there's actually no heterotacky. And I've inferred my ghost model with 2 classes. Okay. And what I can actually do is calculate the probability of every site belonging to either class 1 or class 2. When I say belonging, I mean, what's the probability that this site evolved under class 1 compared to class 2? And these are the inferred classes that we've inferred from the data. Now, it's not hard to kind of think that because there's no heterotacky, class 1 and class 2 are actually the same. And so the probability of being inferred under class 1 is pretty much a linear function, a straight line all the way along. But let's see what happens as we start to introduce some heterotacky. So if we just introduce a little bit of heterotacky, okay, so just by having one branch different between the trees that we simulate to create gene 1 and the tree that we simulate to create gene 2, only one branch is different. What we see is the probability, if there was a lot of heterotacky, we would expect the probability of evolving under class 1 to be higher for the sites that actually did evolve under class 1, and it would be lower for the sites that didn't. So these sites, the first 100, did evolve under class 1, and we see that we start to just increase a little bit. So it's just slightly higher. Just excuse me. Stop. You both have to be very quiet. Stop playing and stop being silly. Daniel, put that all away. Really sorry, everybody. So you can see this line has just increased a little bit. And if we add a little bit more heterotacky, what happens? Okay, sweetie, sit down. What happens if we had a little bit more heterotacky? Well, these are only just one single data set of only 100 sites each class, so there's a lot of noise. So it's maybe gone up just a little bit. Now let's add a bit more. If we get to 20%, all right, we start to see now we're now changing 5 out of 25 of those branch lengths, so we can start to see it really make a difference for class 1. The probability of evolving under class 1 is getting quite high, at least in that first, where it actually did evolve under class 1. And if we keep going, whoops, wrong way, 40% gets even higher, 60%, it's about the same. And actually, I think in this sort of simulation, 80% actually dropped down again. And then maximum heterotacky is right up there. So you missed the first slide, buddy. I've already explained what heterotacky is. Now, if we want to remove this noise, we can just do it 100 times and average over it. And that gives us a nice plot where we can see this sort of effect. So the thing, the direction I'm leading with this is to wonder if a petition model was actually appropriate, that is gene 1 and gene 2 were, the signals were wholly contained within those genes and they were completely separate, then it would be fine. And this is the sort of thing we would see, okay, when we fit a ghost model. But imagine if it's not appropriate, if perhaps the signal or the class is maybe there's a particular domain that's within a couple of genes that's very similar, and it doesn't just fall neatly sort of to this gene boundary. So I actually just permuted the data. So all I did here was just change the order so that class 1, the 100 sites we had for class 1 no longer go from 0 to 100. They're now in this sort of section here, sort of, I think that's about 20 to 70. And then in this section here, and then class 2 is the other bit. So we can see when the site did evolve under class 1, it increases quite steeply. And when it didn't, the slope is quite low. Okay. So now I think, oh no, I have one more thing that I wanted to throw in. And actually this was done a couple of years ago while I was in Vienna. And I think Tanya Hofer actually did these simulations, but I just happened to find the figures while I was creating this talk. And I thought, oh, that actually fits quite well. So we did these simulations where we simulated data with 4 classes. Just excuse me one moment. Daniel Crotty, can you please turn that television off? Off. And so we did it with 4 classes. And what we were trying to simulate here was basically 3 genes, that's red, green, and blue, but some common domain, or not a common, but a domain that was kind of present in all 3 genes and under similar sort of selection pressures. So there was a common phylogenetic signal between these black areas. So we actually have 4 different signals, but only essentially 3 genes. Now, ignore the fact that I've replicated that the second time. So this is how the data was simulated. Then what we did was we fit a ghost model with 4 classes, and for every single site, we colored it red, blue, green, or black, depending on the highest probability. So we looked at the probability distribution for every site of which class did it come from, and we colored it appropriate. And this is what we saw. So as you can see, it's not going to be perfect for every site. There's randomness involved. There's going to be some sites that evolve sort of that are under the red class, that just happen to end up with site patterns that were more likely to have come from the black class or the green or the blue. But you can see the signal is still highly recoverable. We can still see the pattern if we have a look at it. Okay. Now, let's move on to something interesting, which is what do we actually see in real data? All right. So this is a data set that I presented last year, but I didn't show these particular figures. This is a alignment of about 29 cassava brown streak viruses. And just to bring you up to speed, there's 2 recognized strains of cassava brown streak virus. There's this orange one, which is Ugandan cassava brown streak virus. And the green and blue are actually, you can actually ignore the fact that they're 2 different colors for now. They are actually recognized as, oh, I've gone way over time, sorry. I'm going to skip this example.
You've got a couple of minutes.
Just do the next example that I think is the most interesting one. Okay, so this is another data set that I analyzed in the Ghost paper. And I wasn't actually looking for anything to do with the platypus, but I did notice this class, which looked very interesting. It only has a weight of 0.03. It's a very small class, but all species are basically conserved except for the platypus. All right. And I thought, well, that was quite interesting because I started thinking, well, okay, not too unreasonable. The platypus is quite a unique animal. But then I looked at the other animals that we have in this data set. So in addition to platypus, we have 4 different types of turtle or tortoise. We have a couple of crocodilians, alligator and caiman. We have a finch. We have a jungle fowl, a python, a couple of different lizards, a mouse, an alleged human, a lungfish, and some sort of frog or toad. So this is an incredibly diverse thing, an incredibly diverse data set. And it wasn't so much surprising that there was this signal of the platypus diverging from all these others. It was more surprising that the signal said that all the other 15 species were basically conserved in these areas. So in other words, the human, the frog, the lungfish, and everything else that I just showed you are all basically conserved in these areas, but the platypus is doing something quite different. So I actually plotted this data set is 180,000 sites and 250 genes. So I plotted this class, the probability of belonging to this class cumulatively, all the way along from site 1 to site 180,000. And so what I'm looking for here is sections where it gets very steep, because where it gets very steep tells us there's an area here where things are happening to the platypus. The platypus is evolving and the others are not. So I thought that this might be somehow of interest. And so I found some of these areas and some of the genes that were in these areas. And I think this is the steepest one. That red line is the slope that this line should be going at, all things being equal. All right. And you can see it's much, much steeper. So certainly whatever gene that is, and there's the gene number there, there's something kind of strange going on with the platypus compared to all those other species. The next one is also interesting because it shows you that it's not just a signal coming from the whole gene. You know, that there's overall, yes, the gene is steeper, but for the most part of the gene, it's actually climbing sort of slower than the red line. And it's just this section of, who knows, maybe 100 sites or something in the middle there where it's really, so there's some real strong evolution going on with the platypus there. I did start to think, what's so different about the platypus to all these other things? And it did occur to me that the platypus, even though these animals are also very different, the platypus was the only venomous one. And venom is kind of sort of a venom gene will kind of evolve from another gene that non-venomous animals typically possess, and will evolve to do something, to create some sort of venom. So I found this paper which identified 83 platypus venom genes, And I went through those 83 genes, and I went through my my data set of 250 odd genes to find hopefully lots of Venom genes, and they were hopefully going to match. Unfortunately, I only found one. There was only one of these 83 recognized Platypus Venom genes that was in the data set. But here it is. And I was initially disappointed, thinking, oh, well, it doesn't actually climb much higher than the red line you would expect it to. But what it does show is there is still a section, and it's a very small section of 30 to, I think it's about 50 odd sites or something like that, where there's really some serious evolution going on in the platypus, but not any of the other species. So that's pretty much where I'll wrap it up. Yeah, I'm happy to do questions later if I've cut too much into the next person's time, but thank you very much.
Thank you very much. I think, first of all, well done on maintaining a coherent talk whilst parenting.
I'm glad it was apparently coherent.
Best example I've seen of that. I think in the interest of keeping roughly to time, let's hold off the questions, and perhaps that'll give you a little bit of a break, Stephen. And we'll come back to, if there are some questions for Stephen after the next talk in the general discussion, please bear them in mind for then.
Or put them in the chat.
Or put them in the chat, and we'll see them afterwards. Thank you, Venta. And so the last live talk, remember the discussion is going to be slightly different, will be from Sean Buckley. So Stephen, if you would, I think you're not sharing anymore. Yes, you are.
Oh, yeah, that's the problem.
And then Sean, if you can start sharing your screen, then we'll be ready to go.
Yep, so how's that?
Also from Adelaide. So take it away, Sean, on long-term climatic stability drives accumulation and maintenance of divergent lineages in a temperate biodiversity hotspot.
All right, thanks. Yeah, I know the title is a bit of a mouthful, but that's what it is. So hi, everyone. My name's Sean Buckley. I'm currently in the limbo, I guess, between submitting my PhD in August this year and graduating sometime early next year. So my thesis was generally on the phylogeographic history of a group of small freshwater fish, but for the sake of time today, I'm just going to talk about one chapter in particular, focusing on the biogeographic patterns in a temperate biodiversity hotspot. So I should mention that I was a student within the molecular ecology lab at Flinders University in Adelaide, where we've managed to absolutely smash our lockdown, so that's been great. So yeah, I'll just get straight into it. So the biogeographic hotspot in question for this is Southwest Western Australia. Quite a few people might be familiar with this idea that it's quite a unique part of the world. So this is a temperate bioregion in exactly where you would expect it to be, in the southwest corner of the continent. And it's sort of defined by a relatively simple topography and environment that's been relatively stable over history, so over sort of millions of years timescale. So this map on the left here shows sort of the elevation across the region, so generally it's fairly simple. This second map shows precipitation. So this is actually the main factor that defines bioregions within the area. So what we have in Southwest WA are sort of these kind of distinct zones defined by rainfall. So we have the high rainfall province towards the coastline, transitional rainfall province further inland, and then the arid zone for inland. So what makes it a great place to study is it's actually the first of only 2 globally recognized biodiversity hotspots in Australia, and as the first one that was ever defined for Australia. So this is based on the observation of over 8,000 plant species in the region, over half of which are totally endemic to the region. This is a similar pattern in animals, but not quite to the same degree. So the model species group that I work with are called Pygmy Perch. So they're a group of small, endemic and poorly dispersing obligate freshwater fish. So there's a few different species that I'm showing here. They're found in the 2 separate isolated temperate regions of Australia, so in the southwest and the southeast. So there's a total of 7 described species of pygmy perch, but previous genetic work shows there might be at least 10 cryptic species, or 10 species total, including cryptic species. As I said, they're found across these 2 isolated temperate regions, and they're a really good model for studying the evolutionary impact of historical environmental changes because of these characteristics. So they're really reliant on habitat. They can't disperse and easily colonize new areas or anything like that. So they retain the signal of environmental change really well. So for this chapter of the work, I focused on two main species. So we have the Western Pygmy Perch, Nanoperca vittata, who's this very colorful guy here, and the little Pygmy Perch, which sounds a bit redundant, but it actually is smaller than a Pygmy Perch normally is. And that's this quite pale one here. So interestingly, Nanoperca pygmaea, the little Pygmy Perch, was actually only fairly recently described in 2011. This is based on morphological differences, so the number of spines, but as well as other genetic information such as allozymes and mitochondrial data. So previous work that I did during my honors using genomics data suggested that Nanoperca vittata is actually a species complex of up to 3 cryptic species. So that's what's shown in this phylogeny here that shows 3 different lineages that are quite well isolated, and that was supported by species delimitation modeling as well. So one issue with this is that either the previous identification of cryptic species is either based on somewhat limited genetic data in the terms of mitochondrial DNA or allozymes, or fairly limited sampling, so only having a few populations across the region. So sort of the main aims of this chapter was to more thoroughly investigate the identity and nature of these cryptic species, and learning, of course, this is a phylogenetic conference, so a big part of that is determining the evolutionary relationships of those cryptic species, but also trying to look at some functional differences between those species to see what actually makes them unique species. And sort of the overarching aim of this whole section was looking at factors that influence speciation and how that might play a role in the formation of this area as a biodiversity hotspot. So this all feeds into sort of understanding the role of climatic stability, since we know that this has been a fairly stable habitat for a long time, and how this might influence the maintenance of cryptic species and the overall accumulation of biodiversity. So I'm just going to give a very brief overview of my methods pipeline. It's quite extensive, and I'll go over the relevant sections in more detail later on. But within the lab, we use a genomic sequencing approach called double digest restriction associated DNA sequencing, that's a mouthful, to collect thousands of genomic loci across all of our samples. We then do our own in-house bioinformatics and cleaning and filtering of the data. So then I pipe this into sort of 3 different sets of approaches. So the first is sort of a generally descriptive approach. This is looking at genetic distance, fixed differences, and PCA of these populations to just try and get an idea of exactly what level of quantitative genetic difference we see between populations and species. So some more traditional phylogenetic approaches, so predominantly using maximum likelihood. And then some more multi-species coalescent-based species delimitation modeling, so estimating species trees, species delimitation, and that kind of thing. Separate to this, so this is totally without any genetic data, I also used species distribution modeling to map the distribution of these species over time and then project them back into the past. Sort of as a final step, try to combine some of these components together to look at how the species delimitation and the quantitative genetic differences might relate to one another. So this map just shows the sampling design for this project. So all of these samples were collected from museum samples, not fieldwork, which is always a bit disappointing, but you get what you get. So we had a total of 9 populations of both of these species, with around 2 to 5 individuals per population, depending on what kind of samples were available. So we have a total of 7 populations of Nanoperca vittata, which you can see in the diamonds on this map. So it's the much more widespread of the 2 species, and 2 populations of Nanoperca pygmaea in the stars down here. So that actually more or less represents the full distribution of that species. It's really only found in a handful of rivers and is one of the most endangered freshwater fish in the country. So in total, we had 33 samples, and the genomic sequencing approach returned a total of 19,000 rad loci. And from that, we had 18,000 single nucleotide polymorphisms that were unlinked and biallelic, which were used for some of the analyses later. As this is a general overview of what the descriptive patterns found. So in this matrix on the left, the numbers demonstrate pairwise genetic distance between each of these populations, and the colors refer to the number of fixed differences between those populations. So this is when we have one allele that's fixed for one variant in one population and the alternative allele entirely fixed in the other population. So we can consider this as different as a particular locus can be for these 2 populations. And then on the right, we have a PCA of those different populations based on those 18,000 unlinked SNPs. So a pattern that you might see fairly kind of clearly within the matrix is that there are sort of vaguely 3 distinct clusters that we can see in here, where we have smaller difference within the groups compared to differences between those groups. They correspond quite well to the PCA. So what this PCA shows is that there actually are two very distinct clusters of Nanoperca vittata, and that this might relate to cryptic species, but obviously we have to go into that in more detail. So one of the good ways to do that is to look at a maximum likelihood phylogenetic tree. So I did this using RAxML with concatenated radloci, but I also tried partitioning the concatenation by each locus separately, as well as estimating individual gene trees and then summarizing them. All of them returned the exact same results. So what you can see of this tree is that we have really strong hierarchical structure across the clade. Most divergences are supported by 100% bootstrap support, no matter what approach was used. And then there are kind of three main distinct lineages. So those relate very well to those three clusters that we saw in the descriptive group. So that's showing likely that we have two different species of Nanoperca vittata and then Nanoperca pygmea separate. But in order to validate that, I decided to take a more statistical approach. So using SNAP to do species tree estimation and delimitation. So this figure just shows that exact same maximum likelihood tree again on the left, and on the right is kind of a summary heat map of the delimitation scenarios. So each column represents one scenario that was tested. The rows correspond to the samples that are in the tree in the alignment, and then the color is which are considered the same species. So for example, in S2, we can see two different species, either in the light blue or the dark blue, all the way down to S10, where there are 9 different species in total. So because this could be quite analytically intensive to have so much data, both in number of samples and number of SNPs, I chose to subsample it down randomly to the 2 samples with the most amount of data per population and 5,000 randomly selected SNPs, and then evaluated the best delimitation scenario using AICM. Although we've discussed already some of the issues with using AICM in phylogenetic-based approaches, this was again kind of limited by computational ability and time more than anything. And I then extended this to include divergence time estimation by calibrating on previous divergence estimates from that genomics work that I did during my honors as well. So here are some of the results from that. So interestingly, species delimitation actually supported the most possible number of species within the lineage. So it suggested that there were 9 species of Pygmy Perch, but there was kind of an asymptote around 5 species within the modeling. So we know that the multi-species coalescent can overestimate the number of species, particularly when we have very divergent and highly structured lineages within the unit. It tends to see those divergent populations as fully reproductively isolated species, which might not be the case. So we suggest instead that the 3 species that were shown in those other methods is the most conservative estimate. We're going with kind of the lumper approach to it, and that the S5 might actually represent evolutionary significant units within that. And that's kind of supported by the divergence estimates, where again, you can see those 3 lineages. They're very well isolated. Most of them have a divergence time of over sort of 3 million years, which we would think is more than enough time to form reproductive isolation, and that there's a little bit of structure within this more widespread Nanoperca vittata lineage, but not within the other 2. And as you can see, also some of the populations have diverged very recently, so we wouldn't actually think that they are separate species. As a complementary to this, we want to look at how this might relate to environmental changes in the region. So we used an ensemble species distribution modeling approach within Biomod 2. So what this does is it takes occurrence data. So we had 114 points that were taken from the Atlas of Living Australia, as well as prior work, and correlated them with 9 different bioclimatic variables that summarize various aspects of temperature and precipitation across the region. I used 5 different modeling approaches, which were all averaged together to form an ensemble per time period, and then projected those back to various time periods throughout the Pliocene and the Pleistocene, so all the way back to about 3.3 million years ago. So here's just some of the results of that quickly. So this is the ensemble distribution model per time period. So the top left, we have the current distribution that was estimated all the way through sort of the Holocene, the Pleistocene, and all the way back to the Pliocene. So you might notice it's actually, it remarkably looks quite static over time. It doesn't change very much, except for a kind of a coastward expansion during glacial maxima, lower sea levels, but that suitable habitat has always been fairly close to the coastline. And that's well supported by these other two maps, which is, so this top right one in the blue is just the average across all of those time periods together, which shows consistent habitat along the coast. And this pink one's the standard deviation, which doesn't show much variation at all. So the last sort of component of the study that I want to talk about is looking at a functional annotation approach to those fixed differences. So under genetic drift alone, we might expect that fixed differences between populations or species should be spread fairly randomly throughout the genome. So they shouldn't have an association to particular adaptive functions or anything. So what would happen if we find certain functions are overrepresented or enriched, that they might actually be related to some form of selection. So whether this is prior to divergence or not, we can't really say, but it might be associated with it. So this is just a general overview of how we do this. So first we determine where these fixed difference SNPs are. So this example shows a fixed difference for the first species, the second species, and the third species. We obtain the full rad locus sequence for each of those SNPs, so around 100 base pairs. And then we align these to the southern Pygmy Perch genome, so another species within the genus that we already have a genome for. It's the only other Pygmy Perch with a genome, and annotate the functions of those genes. And then doing a gene ontology enrichment analysis to see whether some functions are overrepresented by that. So all these steps are repeated 3 times, once for each of those species, as well as using all of those 19,000 rad loci as the background so that we know when certain functions are actually overrepresented. So there's a lot of information in this table that doesn't really need to go into much detail, but we had fairly good alignment of these loci to the genome. So around 80% of loci were successfully aligned. And a total of 25 gene ontology terms were enriched across all 3 species. So 12 in one in another, and 12 in the third one. And then these Go terms were driven by 96 unique genes. So some genes came up with more than one Go term that was enriched. So this is just a quick overview of the results.
Sean, you have very little time.
Okay, I'll be quick. So this is just an overview of those enriched functions. So interestingly, we found within Nanoperca pygmaea that there were functions related to metabolism, which is makes sense because it has smaller body size, probably related. But also interestingly, chromosome segregation and cytokinesis functions were enriched. And that's interesting because we also found that within one cryptic species of Nanoperca vittata, when we delve in more into the details of that, and also within the other cryptic species of Nanoperca vittata. So it seems like a really important function driving divergence. So just as a caveat, this is intended only to be an exploratory approach. So there are obviously some limitations, the standard ones for doing rad-based work. So how much of the genome is covered, how accurate your ecological annotations are, and of course, actual experimental validation of those results. And also that it's very conservative because fixed differences are about as strongly differentiated as alleles can be between populations, but might not be capturing some of the finer differences. So we have suggested some future extensions which might help address some of these problems. As just a quick summary, so there was very strong hierarchical phylogenetic structure across these populations and species with a lot of fixed differences. It represents at least 2 cryptic species of the Western Pygmy Perch, and this is probably facilitated by having a stable distribution over a long time. So these divergent lineages were maintained throughout the speciation process. So the enrichment of those functions were probably related to how chromosomes are arranged during meiosis, which might have a strong role in reproductive isolation. But of course, we need further developments to be able to improve this method. Of course, there's no real substitute for ecological or experimental data validation, so that might be a more helpful way to validate some of these results. So I'll just skip over this part pretty quickly, but as I said, climatic stability seems to be a really important factor for maintaining this cryptic diversity and is an important factor for these biodiversity hotspots, which are likely going to be threatened by contemporary climate change. As of course, I'd just like to thank my supervisors and all the members of my lab for any help with it, and I'd be happy to take any questions about the project. Thanks.
Thank you, Sean. And before we move on to questions, let's thank Sean and all of the rest of the speakers for this session again. Now, Sean can take a breath. In the interests of getting more or less on time, I suggest that if you have some questions for Sean, for Constantine, Chin, Stephen, or for Nick and Jonathan, you start queuing those up. And I think we're going to hear first from Nick, who will give us a quick overview of his pre-recorded talk, which I'm sure you've all watched. And then Jonathan will do the same. So just hand over to Nick now. Thank you.
Thanks, Mike, for that. So I hope you all saw my amazing talk. So basically what I went through was I studied lots of viral ecology, phylogenetic ecology. And what I did was looking at phylogenetic structure in something that's impacting all of us at the moment, SARS-CoV-2. So basically, being a viral ecologist, when SARS hit, I was obviously interested in Mike, working with Mike. He was like, yes, let's just focus on SARS-CoV-2 for a bit. And rapidly, we use something called GISAID, which is a free online data repository, which has over 200,000 SARS-CoV-2 genomes, which is amazing. We use this resource to start looking for phylogenetic structure within SARS-CoV-2 across the world. And we rapidly found 3 distinct lineages, which don't necessarily have any biogeographic explanation, maybe one, but mostly they're 3 distinct lineages. 2 which were ancestral, which co-circulated in China early on and came to Australia via the Ruby Princess. And then later on, there was, in the first wave in Europe, there was potentially a new lineage called lineage 3, we call it, which exploded with some really neat non-synonymous mutations. And this rapidly spread across the world, and now is the most common lineage you'll see anywhere. We use some pretty cool tools to get at this particular structure, looking for different patterns of coalescence through time. Then we also looked at the population diversity, or sorry, the growth rate of population size through time using phylogenetic methods, and we saw some interesting patterns which I described briefly in the talk. So, is there any questions about any of that? Any queued up?
I don't know if they're already queued up, but Jonathan, do you want to give us a talk about yours and then see if there's
any questions
anything on there as well? Anything yet? Yeah.
Okay, firstly, thanks to the previous talkers, especially Chin, because her talk explained the AIC quite a lot, which was good for me. So my talk, it's called To Be IC or Not to Be IC. It's actually just as much about the AIC as the BIC. I just wanted an interesting title so that hopefully people would watch it. So when you're using the AIC or the BIC, there's a lot of mathematical assumptions that come into deriving the formulae. And often in real world examples, those assumptions actually are violated. So some of the assumptions are pretty weak, others are a lot stronger. Yeah, so what we've done is we've looked at how to actually generalize the AIC and the BIC so they're more widely applicable in phylogenomics and phylogenetics. But these problems arise in all sorts of diverse fields. So the problem comes with the parameter spaces. So if you have constrained parameters, that actually violates the assumptions that you need to derive the AIC and BIC. So if you have a parameter that can be anything from negative infinity to positive infinity, that's fine. But if you have a parameter that can only be positive, like an edge length on a species tree or a substitution rate, that actually violates the assumptions of the AIC and BIC. So this isn't quite technically correct, but it kind of gets the point across, just like how you can think about the effective population size in biology. You might want to look at the effective number of parameters when you use the AIC or the BIC. So our example models are in phylogenomics. So we've got the multi-species coalescent model, which models incomplete lineage sorting. And we look at our generalized AIC and BIC on species trees compared to networks. So the the formula are pretty complicated, so they really only work for small models. Fortunately, our models are small, but you can also, I didn't have enough time to talk about this in the actual talk, but you can find bounds on the, so a lower bound and an upper bound on the penalty term for the AIC and BIC, and you can use them for more complicated models, or you can use a kind of bootstrap to estimate your penalty term if you have a complicated model.
Thanks, Jonathan.
Right, thank you.
Barbara already has a question for you, Jonathan, and also Chip, so maybe if you stay on.
Right, I can kick off with that question. Yeah, Jonathan, thanks for your recorded talk. I really enjoyed listening to that. And Chin, thank you for your talk. So I have kind of a joint question to you, I guess, which is, so given the sort of, I guess, increasing understanding of some of the issues with using AIC in phylogenetics, and I guess increasing ease of being able to compute approximations to the KL distance sort of fairly directly, do you think should we just abandon the AIC? What do you think?
I haven't really looked at the KL divergence, so other than through the AIC, so maybe that's something I should look at. But yeah, it could be probably a good point.
It's can I just interject quickly? Especially in the discrete case, it's a really good point, Barbara, because calculating the KL divergence is really no harder than calculating the AIC or the BIC. In the continuous case, it's a bit harder, but you know.
That's Tristan, everybody.
Yeah, sorry.
It should also be a mic.
Oh,
What do you think, Jim?
Barbara.
No, I don't think so. Sorry, I got echo because I'm using Albert's Zoom. I think sorry. I did 3 simulations. One of the simulation, the tree has no short internal branches. I think that one AIC behaves quite well. I actually got a question for you, Jonathan. What are the assumptions for AIC? It probably is a dumb question. I mean, does it really have any assumptions for AIC?
So most of the assumptions are pretty mild. So basically you have to assume that your random variables are approximately normally distributed. So the central limit theorem will ensure that most of the time. So yeah, and there's not really a lot. So you need to have yeah. So like I said, you need to have parameter spaces where there's no constraints on the parameters. Yeah, I'm not really sure. I have to think about. There's not a lot of constraints. Not a lot of assumptions really.
Yeah, so I don't really think it's a simple question to say we should abandon AIC or not. I think it depends on the conditions of the data. I'm not sure. You agree, Barbara?
Thumbs up from Barbara.
Next, Barbara had another question for Constantine, I believe.
Yeah. Barbara, you have another question for Constantine. You would to go straight to that next?
Yeah, no, thanks for your talk, Constantine. I really enjoyed that. So, so you had kind of a choice to make of. I've always loved a talk where I learn a new word. So these doculects, and trying to decide should they be grouped in a language or not. I was just wondering, like you made a sort of a choice of a threshold there, and then later on you get this sort of uptick towards the present in the birth rate. So I just wondered, did you investigate if you pick a different threshold for deciding a group of doculects are a language, does that sort of change the shape of that subsequent sort of curve?
Yeah, so imagine we had all the doculects that exist. So, or even further, if we have one sample for every person who lives in Vanuatu, then we would see no drop in birth rate. So we would see constant, or maybe even more extreme, we would see an increase. But as soon as we filter out lineages, we see fewer diversification events close to the present. And it goes gradually. So the more we filter out, the less diversification events we see, or the further back in time these diversification events are that we filter out. So that also concludes then that the drop in birth rate goes further back to the past.
Thanks very much.
Okay. We have, yes, that's on. We have another question from Anna Sarah Silva. Do you want to use your mic and ask your question?
Sure. Just asking Sean which definition of cryptic species he used, because at another meeting I attended, there was a whole symposium on what cryptic species are, and at the end of it, there still wasn't a consensus. So I was just curious.
Yeah, so I think I don't think you're ever going to get a real answer to that question, is my gut feeling. Defining a species, it seems to be the thing that'll plague biology forever, I think. For our case with the study, was sort of the point of the functional enrichment stuff was to try and add another layer of evidence for what we consider actually functionally different species, as opposed to just looking at the multi-species coalescent, which, again, as I showed, it's just sort of picking up population structure, not species, and that's very hard when we have the speciation continuum and things are sort of arbitrarily somewhere along this gradient between being just a divergent population or a new species. So ideally, we'd like to be able to infer reproductive isolation between the species, and that's sort of what we suggested to some degree with the chromosome level differences between the functional enrichment characters of those cryptic species. So what we actually know is that there's pretty much no hybridization between Batata and Pygmy at all in the wild. They never find hybridization, even though they're found in the exact same river together, happily mingling, but they just don't interbreed ever. So we think that a big part of that might be how successful they are at recombining, even if they do breed. That's been shown in Drosophila to be a big factor about how hybrids are no longer viable and why they're different species. So the goal is to try and get to being able to infer reproductive isolation, so we could say biological species. At the moment, it's sort of just based on a phylogenetic species concept more than anything.
Okay, thank you, Sean. I think that's as good as answer I've heard to that question as well. I have a question for Stephen Crotty, if you're around. He's apparently still here, so I'm going to ask my question, which was about the heterotacky factor, which is simply based on the number of edges that are different. It's flipped between models. Would it be informative to, or would it just be a waste of time to start thinking about like a more graduated change or a measure of how the heterotacky is changing between two models? Currently, you would have a change of branch length of, say, between 0.05 and 0.06. If they all flipped, you still have the same heterotacky measure. Is that the next thing to look at? Of course, if he's not here, then he's not going to answer that question.
For the recording.
Yes, for the recording.
Other things going on.
He might have a few other things going on. That's fair. Have I seen any other questions in the chat? Bent, anything else? Are there any other questions anybody would like to ask now live or online? And if not, then we might just call that a day. Give you a moment. Don't be shy. We can't see you.
I've got a question for Sean, actually. That was a really great talk. Did you, so you obviously there's a genome available for that group of species. Did you map to, or?
Just for southern pygmy perch, so a different, one of the southeast species we had a genome at the time, which has now been improved since that work was done. So we're aiming to redo some of it. So we got longer scaffolds that are almost chromosome level now, I think. But no genomes for either Vittata or Pygmea. It's only for the other species, which we actually do a lot more work with.
Thanks, Dan.
Okay. Come back to me. Well, I don't think we have any more questions. We do not. No. In which case, thank you once again for all of our speakers and pre-recorded speakers, I guess, for this session. And Venta or Nick, do you have any announcements? Any housekeeping?
No.
No. Well, what time do we start tomorrow?
In
Hobart time?
11 hours and 20 minutes from now.
In 11 hours and 20 minutes.
That's horrible.
8 a.m. Australian Eastern Daylight Time.
8 a.m. in Hobart, which means some of us are going to go home and get some sleep.
Yeah, that's today for some of you and tomorrow for us.
Yeah. All right. Thank you.
Thanks all.
Mute that.